Welcome back to G Fun Facts Online. I am your host, and today we are going to embark on a journey that doesn't take us to the outer reaches of space or the bottom of the ocean, but rather inward, into the deepest and most overlooked territories of our own biology. For centuries, we have operated under the assumption that the map of the human body was finished. We thought we had charted every continent. We knew the heart, the liver, the lungs. We had mapped the rivers of the veins and the arteries. We had identified the microscopic cities of our cells and the telegraph lines of our nerves. Anatomy, for all intents and purposes, was considered a dead science, a subject of memorization, not exploration. We thought we knew everything there was to know about the architecture of us. But imagine for a moment that you have been walking through a dense forest for decades. You have studied every tree, every leaf, every stone. You have cataloged the moss and the insects. And then, after years of this, you suddenly realize that you had completely ignored the air itself. You had missed the medium that held it all together. This is exactly what happened in the world of medicine very recently. In 2018, a team of researchers stumbled upon something that had been hiding in plain sight for the entire history of human medicine. It wasn't a microscopic particle. It wasn't a rare genetic mutation found in one distinct population. It was a massive, body-wide structure, a fluid-filled superhighway functioning beneath our skin, surrounding our gut, encircling our lungs, and sheathing our muscles. They called it the interstitium. This discovery didn't just add a footnote to medical textbooks, it threatened to rewrite them entirely. It suggested that what we previously dismissed as dense, boring connective tissue was actually a dynamic, living network, a shock-absorbing, fluid-transporting, immune-regulating system that might just be the largest organ in the human body. So, for the next 20 minutes, I want to tell you the story of that discovery. We are going to dissect the anatomy of this hidden web, explore how it fundamentally changes our understanding of cancer and healing, and even look at how it might bridge the gap between Western science and the ancient mysteries of traditional Chinese medicine. Let's start with part 1, the accidental discovery. The story begins in 2015 in an endoscopy suite at Mount Sinai Beth Israel in New York City. Dr. David Carlock and Dr. Petros Benias were performing what should have been a routine procedure. They were examining a patient's bile duct, looking for signs of cancer spread. But they weren't using the standard tools of the trade. They were using a cutting-edge piece of technology called probe-based confocal laser endomicroscopy, or PCLE. Now, this is important because unlike traditional microscopes that require you to slice a piece of tissue out of the body, stain it, and put it on a glass slide, PCLE allows doctors to peer into living human tissue in real time. It involves inserting a microscopic camera probe directly into the body to see the tissue while the patient is still alive and the blood is still flowing. So they are looking at this patient's bile duct on the monitor. According to every anatomy textbook written in the last 100 years, the wall of the bile duct should be a dense, solid wall of connective tissue. It should look like a brick wall. But the live feed showed them something else entirely. They saw a bizarre pattern. It looked like a honeycomb. It was a lattice of dark, open spaces, supported by a mesh of thick, bright bands. It looked like a series of fluid-filled cavities. Confused, they checked their equipment. They checked the literature. There was no record of such a structure existing in the bile duct or anywhere else. They decided to take a sample of this tissue and send it to Dr. Neil Thies, a prominent liver pathology at NYU Langone Health. But here is where the mystery deepened. When Dr. Thies looked at that same tissue sample under a standard microscope, the kind used in pathology labs all over the world, the open spaces were gone. The honeycomb had vanished. The tissue looked exactly like the dense, solid wall described in the textbooks. This discrepancy sparked a detective story that unraveled a century-old error in medical observation. It turned out to be a problem of methodology. Dr. Thies realized that the standard method of preparing tissue for study was effectively destroying the evidence. You see, for decades, pathologists have fixed tissue samples. This involves dehydrating them with chemicals and embedding them in wax to make them stable enough to slice. This process creates a perfect snapshot of the cells, but it drains all the fluid. Dr. Thies hypothesized that the dense connective tissue seen in textbooks was actually a collapsed building. In the living body, these tissues were open, fluid-filled skyscrapers. But when the fluid was drained during the fixation process, the floors, which we now know are collagen bundles, collapsed on top of each other. They appeared as a solid, flat pancake of tissue on the slide. For years, pathologists had seen tiny cracks in these slides, but they had dismissed them as artificial tears caused by the slicing process. It turns out, those cracks were the footprints of the vanished fluid. The open spaces were real. They had just never survived the journey to the microscope slide before. To prove this, the team changed their protocol. They froze tissue samples immediately after removal, preserving the fluid structure before the collapse could occur. And there it was. Under the microscope, the honeycomb structure remained. The phantom was real. In March 2018, They published their findings in scientific reports. They proposed that this network of fluid-filled spaces was not just a quirk of the bile duct. They found it everywhere. In the dermis of the skin, the lining of the digestive tract, the lungs, the urinary system, and the fascia between muscles. They argued that because this network possessed a unitary structure and function, it should be classified as a distinct organ, the interstitium. The announcement sent shockwaves through the scientific community. Headlines blared that scientists had discovered a new organ. And while some anatomists debated the semantics of the word organ, no one could deny the physical reality of what Thies and his colleagues had illuminated. The body was not a collection of solid parts packed in inert filler, it was a fluid continuum. Now that we know it's there, let's move to part 2, the anatomy of the hidden web. To understand the interstitium is to understand the architecture of life itself. It is the space between, but it is far from empty. Imagine a sponge. The solid material of the sponge is a lattice built from 2 primary proteins. First, you have collagen bundles. These are the pillars of the system. Thick, strong bundles of collagen. Types Y, 3, and V form the walls of the fluid compartments. They provide tensile strength, ensuring that our tissues don't simply dissolve into a puddle. Interwoven with the collagen are fibers of elastin. As the name suggests, these provide elasticity, allowing the lattice to snap back into shape after being compressed or stretched. But unlike a kitchen sponge, this biological sponge is incredibly organized and spans the entire body. Filling the spaces between these protein bundles is the interstitial fluid. For a long time, scientists knew this fluid existed. It is the nutrient-rich broth that bathes our cells, but they thought it merely seeped passively through dense tissue, like water through soil. The new discovery revealed that this fluid actually flows through open highways. It is a dynamic river system. The interstitium contains roughly 20% of the body's total fluid volume. That is more than the amount of blood in your veins. This fluid is not stagnant. It is in constant motion, generated by the leakage of blood plasma from capillaries. It flows through the interstitial lattice, washing over cells, delivering nutrients, and collecting waste. Eventually, it drains into the lymphatic system. This led to a crucial realization. The interstitium is the source of lymph. It is the pre-lymphatic system. Before immune fluid enters the lymph nodes to be scrubbed of bacteria, it travels through the open roads of the interstitium. So why does this structure exist? What is its evolutionary purpose? The primary function appears to be mechanical protection. The interstitium is found in tissues that undergo constant rhythmic movement. Think about the pulsing of your arteries, the peristalsis of your gut, the expansion of your lungs, or the compression of your skin. If these tissues were solid and rigid, the constant friction and pressure would tear them apart. The fluid-filled interstitium acts as a body-wide shock absorber. Because water is incompressible, the fluid-filled spaces prevent the tissues from collapsing under pressure, while the collagen lattice prevents them from overexpanding. It is a perfect hydraulic cushioning system, protecting the delicate machinery of our organs from the wear and tear of daily life. However, this open highway has a dark side. This brings us to part 3, the superhighway of disease. The discovery of the interstitium was not just an anatomical curiosity. It provided the missing link for understanding how diseases, particularly cancer, spread with such terrifying speed. For decades, oncologists were puzzled by the behavior of certain cancers. A tumor might start in the stomach, but metastatic cells would appear in lymph nodes far away, much faster than expected. If the connective tissue was a dense wall, as we previously thought, how were these large cancer cells tunneling through it so quickly? The interstitium provided the answer. The connective tissue wasn't a wall, it was a highway. When cancer cells break away from a primary tumor, they don't have to hack their way through dense muscle or fibrous tissue. They simply slip into the interstitial space. Once inside, they are in a fluid-filled channel with little resistance. Dr. Thies described it chillingly. He said, once they get in, it is like they are on a water slide. The fluid flow within the interstitium carries these malignant cells directly toward the lymphatic system. This explains why the presence of cancer in the stroma, or the connective tissue layer, is such a critical prognostic factor. It marks the moment the enemy breaches the castle walls and enters the river system that connects the entire kingdom. Recent research from 2020 to 2025 has deepened this understanding. Scientists found that cancer cells don't just passively float, they actively navigate this space. Some cells ride the natural downstream flow toward lymph nodes, a process known as autologous chemotaxis. Surprisingly, some aggressive cancer cells can even crawl upstream, against the flow, using the stress of the fluid on their membranes to guide them to other tissues. This knowledge is revolutionizing cancer staging. It suggests that sampling the interstitial fluid could provide an early warning system, detecting metastatic cells before they even reach the lymph nodes or the blood. Beyond cancer, the interstitium explains the mechanics of other diseases as well. Take edema, or swelling. Edema is essentially the flooding of the interstitium. When the balance of fluid entering and leaving these spaces is broken, perhaps due to heart failure or kidney disease, the rooms of the interstitium overfill, leading to visible swelling. On the other side of the spectrum, we have fibrosis, or scarring. In diseases like scleroderma or liver cirrhosis, the delicate collagen lattice becomes corrupted. The fluid spaces collapse, and the collagen bundles thicken and fuse. The shock absorber turns into a stiff, unyielding scar, causing the organ to fail. Now I want to pivot to something that I find absolutely fascinating. This discovery resonates well beyond the walls of Western hospitals. Part 4. The Eastern Connection. Perhaps the most fascinating ripple effect of this discovery lies in the ancient clinics of traditional Chinese medicine, or TCM. For over 2,000 years, acupuncturists have treated the body based on a map of meridians, channels through which qi or energy flows. Western anatomy has notoriously failed to find physical evidence of these meridians. If you dissect a human arm, you find nerves, veins, and muscles, but you find no energy tubes corresponding to the acupuncture charts. Until now. In TCM, there is an organ system called the sanjiao, or triple burner. Unlike the heart or stomach, the sanjiao has no distinct physical sac. Ancient texts describe it as having a name but no form. It is described as a system of spaces and waterways responsible for the movement of fluids and energy throughout the entire body. Does that sound familiar? The description of the sanjiao aligns with startling precision to the description of the interstitium. TCM describes a reticular network connecting all organs. Science finds a body-wide network of connective tissue. TCM says it controls the metabolism of fluids. Science confirms it transports interstitial fluid and lymph. TCM locates it between the skin and muscles and around the organs. Science locates it in the dermis, fascia, and organ linings. Many integrative medicine researchers now propose that the interstitium is the anatomical basis of the sanjiao. This connection may also explain how acupuncture works. Critics have often asked how inserting a needle into the toe can possibly affect the liver. If the body is a collection of isolated parts, it makes no sense. But if the body is interconnected by a continuous, fluid-filled web, it becomes plausible. One theory is mechanical transduction. When an acupuncture needle is inserted and rotated, it winds the collagen fibers of the interstitium around it, like spaghetti on a fork. This mechanical stress signal is transmitted through the collagen lattice, or fascia, at high speeds, potentially reaching distant organs. Another theory involves electrical conductivity. The interstitial fluid is an electrolyte-rich solution. It conducts electricity better than the surrounding cells. The meridians might simply be the paths of least electrical resistance flowing through the interstitial channels. Stimulation of these points could modulate the bioelectric signals passing through this hidden web to the internal organs. While this is still being debated, the discovery has provided the first viable anatomical bridge between the reductionist view of Western anatomy and the holistic view of Eastern medicine. So what does this mean for us moving forward? Part 5. The Future of Medicine. We are currently in the infancy of interstitial science. The recognition of this network as a functional unit opens up frontiers that were previously unimaginable. First, we are looking at a new diagnostic window. Currently, we diagnose disease primarily through blood or tissue biopsy. The interstitium offers a third window. Interstitial fluid, or ISF. Sampling is poised to become the next big diagnostic tool. ISF contains a different biomarker profile than blood. It is the direct environment of the cells. While blood tells us what is happening system-wide, ISF tells us what is happening in the local neighborhood of the tissue. Wearable microneedle patches are already being developed to continuously monitor glucose, lactate, and cancer markers in the interstitial fluid without drawing blood. Second, we have drug delivery systems. Getting drugs into solid tumors is notoriously difficult because the high pressure within a tumor often pushes drugs away. Understanding the fluid dynamics of the interstitium could allow engineers to design nanoparticles that ride the interstitial currents directly into the heart of a tumor. And finally, immunology and healing. We now know that the interstitium is a highway for macrophages and other immune cells. Understanding how these cells navigate the lattice could lead to new treatments for autoimmune diseases. If we can close the road to inflammatory cells in conditions like Crohn's disease or rheumatoid arthritis, we could stop the damage without suppressing the entire immune system. As we conclude this episode, I want to leave you with a thought on the philosophical shift this represents. The discovery of the interstitium is more than just a triumph of microscopy. It is a triumph of perspective. For centuries, Western medicine has been dominated by a part-list mentality. We break the body down into smaller and smaller pieces: organs, tissues, cells, organelles, molecules, assuming that if we understand the parts, we understand the whole. But the interstitium reminds us that connection is as important as composition. You cannot understand a forest just by studying the trees. You must understand the air, the soil, and the water that flows between them. The human body is not a machine made of distinct parts bolted together. It is a fluid continuum. The skin is connected to the fascia, which is connected to the lung lining, which is connected to the heart sac. When you take a breath, the pressure wave travels through this web. When you move your arm, the fascia slides and the fluid shifts, rippling through the lattice. We are a singular, unified, hydrodynamic organism. It is humbling to realize that in the 21st century, in an age where we can map the genome and photograph black holes, we can still discover a new organ inside our own bodies. It begs the question, what else is hiding in the empty spaces? The interstitium was invisible not because it was too small to see, but because we didn't know how to look. We were looking at the bricks and ignoring the mortar. We were looking at the dried ruins and missing the flowing river. As we move forward, the hidden web will likely become a central focus of medical research. It bridges the gap between structure and function, between the solid and the liquid, and potentially, between the ancient wisdom of the past and the high-tech medicine of the future. The map of the human body has changed, the empty spaces have been filled, and we are just beginning to explore the depths of this inner ocean. Thank you for listening to G Fun Facts Online. If you enjoyed diving into the hidden layers of your own biology, make sure to subscribe, and we will see you next time for another deep dive into the wonders of our world.